हेलो फ्रेंड्स, तो स्वागत है आपके अपने यूट्यूब चैनल एवरीथिंग हेयर। फ्रेंड्स, सबसे पहले आप हमारे चैनल पे न्यू हैं तो सामने सब्सक्राइब का बटन है, उसे दबा लें और बेल आइकन को दबाना ना भूलें, जिससे हमारी न्यू वीडियोस या तो कोई भी नोटिफिकेशन आपको जल्द से जल्द प्राप्त हो पाए। फ्रेंड्स, आज हम लोग क्लास 11th का जो चैप्टर 8 है, सेल द यूनिट ऑफ लाइफ, इसको आज हम लोग कंप्लीट NCERT रीडिंग करने जा रहे हैं। तो आइए फ्रेंड्स, हम अपने वीडियो को स्टार्ट करते हैं। What is a cell? 8.1 Unicellular organisms are capable of first independent existence and performing the essential function of life. Anything less than a complete structure of cell does not ensure independent living. Hence, cell is the fundamental structural and functional unit of all living organisms. Anton Leeuwenhoek first saw and discovered a live cell. Robert Brown later discovered the nucleus. The invention of microscope and its improvement leading to the electron microscope revealed all the structural detail of the cell. 8.2 Cell Theory. In 1838, Matthias Sladen, a German botanist, explained a large number of plants and observed that all plants are composed of different kinds of cells which form the tissue of the plant at about the same time. Theodore Swan, 1839, a British zoologist, studied different types of animal cells and reported that cell had a thin outer layer which is today known as plasma membrane. He also concluded, based on his study of plant tissue, that the presence of cell wall is unique character of the plant cell. On the basis of this, Swan proposed the hypothesis that the bodies of animals and plants are composed of cell and product of cell. Sladen and Swan together formulated the cell theory. This theory, however, did not explain as a how new cells were formed. Rudolf Virchow, 1855, first explained that cell divided a new cell are formed by pre-existing cell, omnis cellula e cellula. He modified the hypothesis of Sladen and Swan to give the cell theory a final shape. The cell theory, as understood today, is first, all living organisms are composed of cell and product of cell. Second, all cells arises from pre-existent cell. 8.3 An overview of cell. You have earlier observed cell is an onion peel and or human cheek, cells under the microscope. Let us recollect their structure. The onion cell, which is a typical plant cell, has a distinct cell wall as its outer boundary and just within its cell membrane. The cell of the human cheek have an outer membrane as the delimiting structure of the cell. Inside each cell is dense membrane bond structure called nucleus. The nucleus contains the chromosome, which in turn contain the genetic material DNA. Cells that have membrane bond nuclei are called eukaryotic, whereas cells that lack a membrane bond nucleus are prokaryotic. In both prokaryotic and eukaryotic cells, a semi-fluid matrix called cytoplasm occupies the volume of the cell. The cytoplasm is the main arena of cellular activities in both the plants and animal cell. Various chemical reaction occurs in it to keep the cell in the living state. Besides the nucleus, the eukaryotic cells have other membrane bond distinct structure called organelles like endoplasmic reticulum, the Golgi apparatus, lysosome, mitochondria, microbodies, and vacuoles. The prokaryotic cell lack such membrane bond organelles. Liposomes are non-membrane bond organelles found in all, both eukaryotic as well as prokaryotic. Within the cell, ribosomes are found not only in the cytoplasm but also within the two organelles, chloroplast in plant and mitochondria and on rough endoplasmic reticulum. Animal cells contain other non-membrane bond organelles called centrosome, which helps in the cell division. Cell differs genetically in size, shape and activities. For example, mycoplasma, the smallest cell, are only 0.3 in length, while bacteria could be 3 to 5 meter. The largest isolated single cell is the egg of an ostrich. Among multicellular organisms, human red blood cells are about 7.5 meter in diameter. Nerve cells are some of the largest cells. Cells are very greatly in their shape. They may disc-like, polygonal, columnar, cuboid, thread-like, or even irregular. The shape of the cell may vary with the function of they perform. 8.4 Prokaryotic cells. The prokaryotic cells are represented by bacteria, blue-green algae, mycoplasma, and PPLO. They are generally small and multiply by rapidly than eukaryotic cell. They may vary greatly in shape and size. The four basic shapes of bacteria are Bacillus, Coccus, Vibrio, Spirillum. The organization of prokaryotic cells is fundamentally similar even though prokaryotes exhibit a wide variety of shape and functions. All prokaryotes have a cell wall surrounded the cell membrane except in mycoplasma. The fluid matrix filling the cell is the cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not enveloped by nuclear membrane. In addition to the genomic DNA, the single chromosome circular DNA, many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called plasmids. The plasmid DNA confers certain unique phenotypic character to such bacteria. One such character is resistance to antibiotic. In higher classes, you will learn that the plasmid DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotes. No organelles like one in eukaryotes are found in prokaryotic cell except for ribosomes. Prokaryotic have something unique in the form of inclusion. A specialized differentiated form of cell membrane called mesosome is the characteristic of prokaryotes. These are essentially inflooding of cell membrane. 8.41 Cell envelope and its modification. Most prokaryotes cell, particularly the bacterial cell, have a chemically complex cell envelope. The cell envelope consists of tightly bound three-layered structure that is the outermost glycocalyx, followed by the cell wall and then the plasma membrane. Although each layer of the envelope performs distinct functions, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of their differences in the cell envelopes and the manner in which they respond to the staining procedure developed by Gram-Weiss. Those that take up the gram strain and gram positive, the other do not, are called gram negative bacteria. Glycocalyx differ in composition and thickness among different bacteria. It could be loose sheath called the slime layer in some, while in the other it may thick and tough called capsule. The cell wall determines the shape of the cell and provide strong structural support to prevent bacterium from the bursting and collapsing. The plasma membrane is selectively permeable in nature and interacts with the outside world. This membrane is similar structurally to that the eukaryotes. Special membranous structure is the mesosome, which is formed by extension of plasma membrane into the cell. These extension are in the form of bacillus, tubule, and lamella. They help in cell wall, DNA formation, DNA replication, and distribution of daughter cell. They also help in respiration secretion process to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotic like cyanobacteria, they are are other membranous extension into the cytoplasm called chromatophores which containing pigments. Bacterial cells may be motile on the non-motile. ओके फ्रेंड्स, आगे के पार्ट को हम लोग सेकेंड देखेंगे। थैंक यू।